Hey guys, it's Medicosis Perfectionalis, where medicine makes perfect sense. This is the 25th video in my physiology playlist. We will review cell membrane physiology, which includes diffusion, active transport, osmosis, G protein, Fick principle, and the Donnan equilibrium. So let's get started. So this is my YouTube channel and the playlist is called Physiology. We are talking about cell membrane physiology. The first video was introduction, then 23 videos about the actual subjects, and this is the 25th video review. In the upcoming videos, we will have autonomic nervous system physiology. We'll start with introduction and we'll end with a review. So if you don't have time, if you just watch the reviews, you should be fine. Let me answer the question of the previous video. Here's at equilibrium. They're asking about the Donnan equilibrium. So, Donnan equilibrium, how do you do it? First, we have 6 plasma proteins here. Let's put them in the plasma compartment because they are too big to pass through the membrane. Here you see the chloride and the sodium. And here you see the chloride. And what is the sodium? We have no idea. So, Donnan equilibrium tells us that the product of electrolytes here in the non-protein compartment has to equal the product of electrolytes in the protein compartment. So 2 times x equals 4 times 4. 2 times x equals 16. X equals 8. This is the number of sodium particles in the protein compartment. It's a piece of cake. What does the word physiology mean? Physiologia, origin or nature, study. So it's study of origin or nature. It's the scientific study of the function of a human body and the mechanism of these functions. 60% of your total body weight is water. You have intracellular and extracellular. Which of these is the internal environment? People will say, oh, the internal environment is the intracellular. Shut up. The internal environment is the extracellular. I don't believe you. Imagine a fish that is swimming in water. The water is the environment of the fish. So, the extracellular fluid is the water around your cell. Your cells are literally living inside the ECF fluid. What is physiology? Homeostasis. What is pathology? Lack of homeostasis. So homeostasis is maintenance of not constant because nothing is perfect, nearly constant circumstances in your body's internal environment, which is the ECF. Your cell is made of cell membrane, a nucleus, cytosol, organelles, and don't forget the cytoskeleton. What's the difference among protoplasm, cytoplasm, and cytosol? Protoplasm has the cytoplasm and the nucleoplasm, which is the nucleus and the nucleolus, etc. But then the cytoplasm is cytosol and organelles. Speaking of organelles, the mitochondria is the power plant, lysosome is the soldier, endosome is the delivery guy. In the plasmic reticulum, we have rough and smooth. The rough is the translator, the smooth is the fat synthesis, it's the donut. Golgi is the sorter, peroxisome is the gym trainer, it breaks down fat. Your cell membrane or plasma membrane is made of a hydrophilic head and hydrophobic tail, and it's always a bilayer. Any membrane in your body is a bilayer, 2 layers. Here is 1 and here is 2. Why is the hydrophilic to the outside and inside while the hydrophobic is in between? Think about it, because you have water on the outside and water on the inside, so it makes perfect sense to put the thing that's water soluble or hydrophilic towards water, and the thing that's hydrophobic, which is lipid, away from water. Think. You do not have a cell wall, you just have a cell membrane. Your cell membrane is elastic, dynamic, and semi-permeable, made of proteins, mostly proteins, lipid, and carbohydrates. Speaking of lipid, you have phospholipid, cholesterol, other lipids including sphingolipid, especially in nerve cells. Your cell membrane is a barrier. Your cell membrane is a medium for passage. What is the cell membrane lipid? Phospholipid. And some cholesterol. Tell me about the phospholipid. I have head, I have tail. 1 head and 2 tails. Hydrophilic head, hydrophobic tails. The head has some phosphate, and that's why we have negative charges. If you remember my video about the ESR, yep, those doozy red blood cells had negative charges. That's why they repel with each other, and this was the story of the erythrocyte sedimentation rate. We have 3 types of cell membrane protein: structural, which are the same structure of the membrane, integral, and peripheral. Integral, they take the whole freaking thickness, like this beautiful channel, or carrier, or pump. Integral, they do not take the whole thickness. They are either on the outside or the inside. The receptor to the outside, the enzyme is to the inside. The GPI anchors are examples of peripheral proteins. Check out my video called Paroxysmal Nocturnal Hemoglobinuria. Let's talk about the cell membrane carbohydrate. They are always attached to something. Glycoprotein, carbohydrate attached to proteins, or glycolipid, carbohydrate attached to lipid. Functions of carbohydrate, the ID, so recognition site, adhesion, negative charges, and receptor. On the outside of your beautiful membrane, you have glycocalyx, which is a sugar coat. Your lipid membrane contains phospholipid, cholesterol, and other lipids. Other lipids include sphingolipids, which is sphingosine plus fatty acids. Sphingolipids have a problem in a group of diseases called sphingolipidoses. The GPI anchor protein is a peripheral involved in paroxysmal nocturnal hemoglobinuria. Every membrane in your body is a lipid bilayer. It's lipid. That's why if you're lipid, you can pass through the membrane easily, like a sharp knife through warm butter. But if you're water soluble, man, you can pass but very slowly. It's almost impossible. And that's why we need a channel for you. You can come in through the channel. Because if you would like to try to the lipid, good luck with that. It's possible, but it's very slow. Cell membrane transport, passive such as diffusion or active transport. Diffusion is down gradient. Active is against gradient. What gradient are you talking about? We have two types of gradients. Concentration gradient from high concentration to low concentration, and electrical gradient. Active is against the gradient. We have active carrier transport, primary and secondary, and we have vesicular transport, exocytosis and endocytosis. Cell membrane transport, passive, diffusion, or active. Passive, we have three types: simple diffusion, osmosis, which is a simple diffusion just for water, and facilitated diffusion. Passive diffusion in general, there is no need for ATP. All of them happen along the electrochemical gradient from high concentration to low concentration. Simple, no carrier, through the lipid membrane or through channels, especially if you're small or water soluble, or through both, such as water. Water can go through the membrane very slowly indeed, or it can go through a channel very quickly indeed, such as the aquaporin channel. Osmosis is a special type of simple diffusion just for water. Both simple diffusion and osmosis require no carrier. However, facilitated diffusion requires a carrier. Still no ATP, still along the gradient, but we do require carrier. The carrier will give me three characteristics: structural specificity, like a key in a lock, like a truck in a dock, competition, and saturation by the Vmax. Next, active transport, active carrier transport, and active vesicular transport. Active carrier, primary and secondary, active vesicular, endo and exo. This is the Vmax by the facilitated diffusion because we have a carrier. If we have a carrier, the carrier will have a certain capacity beyond which, I'm sorry. Don't forget that there is a huge difference between obligated water, which is water that follows electrolytes, and free water. Osmosis, simple diffusion of water. No energy needed, no carrier needed. From high concentration of water to low concentration of water, or from low concentration of solute to high concentration of solute. The osmotic pressure depends on the number, not the mass of molecules, the number. So when glucose says, "Oh, I'm angry because I'm heavier than sodium and heavier than chloride, I should be more osmotically active." Shut up, glucose. We only care about the number. Sodium is more abundant than you. That's why sodium wins when it comes to osmotic pressure. Osmotic pressure depends on the number. Osmolarity is per liter, while osmolality is in kilogram. Sodium is the most important. Water follows sodium. Too much sodium on the outside, the cell will shrink. Too much sodium on the inside, the cell is gonna swell. Relatively speaking, make believe that this cell is in your brain, you get mental status abnormalities. That's why sodium problems, CNS problems. Measured osmolality versus calculated osmolality. Measured osmolality, just get a blood sample, send it to the lab, the osmometer will do the work. But calculated, you need to use your freaking brain using this equation. 2 times sodium plus glucose over 20 plus BUN over 3. Normal serum osmolality is 290. You can argue it's 275 to 295. Shut up. Is there a difference between the measured and the calculated osmolality? Yep, it's called the osmolar gap. Normally it should be less than 10. If it's more than 10, Houston, we have a problem. There is a Trojan horse that has invaded your body, a foreign substance such as methanol. Oncotic pressure, we have lots of proteins in your plasma. They will keep the water in because water flows towards the proteins. Lots of water in my vessels, I can perfuse my heart, my brain, etc. What if I have low albuminemia, which is less plasma proteins? I get edema because water will flow to the opposite direction, and accumulation of fluid in the interstitial space is known as edema. What can cause decrease my oncotic pressure? If you're not eating protein, kwashiorkor. If you're peeing protein, nephrotic syndrome or Meniere's disease. Pooping protein, malabsorption syndrome. Decreasing protein synthesis, such as liver disease or plasma loss in third-degree burn. We have two types of edema, a transudate or an exudate. Transudate is a pitting edema, while an exudate is a non-pitting edema. Lymphedema starts as pitting but later becomes non-pitting edema. Factors that affect diffusion through a membrane are very important. You need to memorize these. How does the cell get nutrients? If it's water, it can pass slowly through the lipid or pass quickly through an aquaporin channel. If you're lipid soluble, you can go through the cell membrane. If you're macromolecule, you need spinocytosis or phagocytosis to go into the cell. Vesicular transport is an active transport, therefore it requires ATP. We have two types. Come in, endocytosis. Get out, exocytosis. Here is a comparison between pinocytosis and phagocytosis. The active carrier transport requires ATP. We have primary and secondary. And we have three types: uniport, symport, and antiport. Let's start with primary active transport. How many types? We have four: sodium, potassium ATPase, calcium ATPase, hydrogen ATPase, hydrogen potassium ATPase. Let's start with the sodium potassium ATPase. Sodium out, potassium in. This beloved sodium-potassium pump protects the cell from swelling, and this is why normally we have more sodium on the outside and more potassium on the inside. When you pump 3 positive sodiums out and 2 positive potassiums in, the net result is 1 positive out, which is the same thing as saying 1 negative in. That's why the resting membrane potential is negative. This beloved primary active transport can provide energy for the secondary active transport. Example, look at your heart muscle. It has primary sodium-potassium ATPase and secondary sodium-calcium exchanger. Normally, sodium out, potassium in. The accumulation of sodium on the outside will force sodium to the inside and calcium to the outside. Digoxin will inhibit the primary active transport, and therefore there is no secondary active transport because secondary depends on the primary. When there is no secondary active transport, calcium will not go out. Calcium will stay in. More calcium in, more contraction. And that's why digoxin is an inotropic drug, because it increases cardiac contractility. Another fun fact about pharmacology. Normally, sodium-potassium ATPase, sodium out, potassium in. But if you give beta blockers, they inhibit the sodium-potassium ATPase. Therefore, potassium will stay out, leading to hyperkalemia. But what if, instead of giving beta antagonist, we try beta agonist? Beta agonist will stimulate the sodium-potassium ATPase. More sodium out, more potassium in. More potassium in, this means less potassium out. Yeah, that's why beta agonists can lead to hypokalemia, and therefore they can treat hyperkalemia. If you look at the treatment of hyperkalemia, look at this. Beta agonists, because they stimulate the sodium-potassium ATPase, causing potassium influx, treating the hyperkalemia. How can we boost cardiac contractility? You have many ways of doing it. First, you can give digoxin, which will inhibit primary ATPase, therefore inhibit secondary ATPase. Calcium will not exit, calcium will remain in. Calcium will remain in, leading to calcium-induced calcium release from the sarcoplasmic reticulum. More calcium, more contraction of the cardiac myocyte. There is another way to boost cardiac contractility. Stimulate the beta-1 receptor by giving dobutamine, isoproterenol, or catecholamines. When you stimulate beta-1 receptor, they act as GS-coupled. Oh, what is G protein? G protein is a protein that joins the receptor on the outside to the enzyme on the inside. Cool. Now adenylate cyclase is active. ATP to cyclic AMP. Cyclic AMP will activate protein kinase A, open the calcium channel. Calcium will enter into the cardiac myocyte, go to the sarcoplasmic, calcium-induced, calcium release, calcium contraction. There is another way to boost cardiac contractility by inhibiting the degradation of cyclic AMP, such as milrinone, enamrinone, amrinone. These medications inhibit the phosphodiesterase, therefore cyclic AMP will accumulate, activating protein kinase A. Calcium enters into the cell, calcium-induced, calcium-released, activation, contraction. These drugs is how we can treat congestive heart failure by boosting cardiac contractility. However, calcium channel blockers are contraindicated in CHF. Why? Because calcium channel blockers will block the calcium channel. Calcium will not be able to enter into the cell. There is no calcium-induced calcium release. There is no contractility. And when you have heart failure and I give you something that will decrease your contractility, you should kick me in the nuts. If you would love to learn more about cardiac pharmacology, I have a course consisting of 50 videos, 25 cases, and 25 questions, plus notes, plus a mind map. Go to medicosisperfectionalis.com. So this was the first ATPase known as sodium-potassium ATPase. Now it's time for calcium ATPase. Calcium ATPase, where do you find it? In the muscle cell, in the sarcoplasm. You see that? Calcium ATPase. Yep, calcium, calcium, calcium. Get the calcium out. What if we have no energy, like no ATP? This is not going to work. Therefore, what? Calcium is not going to be able to exit the cell. Calcium will stay inside. So why is that a problem? This is rigor mortis, doofus. This happens when you're dead. The third and fourth type of primary active transport is the hydrogen ATPase and hydrogen potassium ATPase. You find them in the late and collecting tubules in your kidney. Can you give me an example of secondary active transport? Yeah, you know the voltage-gated sodium channels, which we'll talk about later in the action potential? Oh yeah, they enter the channel. Do you see any ATP here? No, I don't, but they depend on the sodium-potassium ATPase. That's why it's secondary active transport. If only sodium is coming in, what do you call this? This is a uniport. What if I'm trying to absorb or reabsorb glucose? Glucose will enter into the cell together with sodium. This is what? A symport, which is a secondary active transport, secondary to the primary sodium-potassium ATPase. For the smart students, there is a huge difference between glucose transporters, the GLUTs, and sodium-glucose co-transporters. Glucose transporter is a facilitated diffusion, but the sodium-glucose co-transport is a secondary active transport. This one does not require energy, this one requires energy. Both of them require a carrier. If you'd like to learn more about GLUTs, I have made many videos about GLUT transporters. Just search in YouTube Medicosis GLUT, which is kind of inappropriate. Okay, here are your ions. Sodium is more abundant in the extracellular fluid outside the cell. Chloride on the outside. Calcium also on the outside. Bicarbonate on the outside. And glucose on the outside. On the inside of the membrane, however, in the ICF, you have lots of potassium, magnesium, proteins, and phosphate. What's your mnemonic, medicosis? Anything that starts with a P. Potassium, proteins, phosphate. How about magnesium? Magnesium is present in the supernova. Oh, yeah. Quick review. Cell membrane proteins are structural, integral, or peripheral. Integral proteins are, like, they take the whole freaking thickness. Channels and carriers. Channels are either gated or non-gated. Gated, ligand-gated, or voltage-gated. These ones open by chemistry. These open by electricity. The ligand, where's the ligand? Could be inside the cell, internal ligand, or outside the cell, external ligand. Another function is the carrier. Could be for facilitated diffusion or for active transport. An example here is the GLUT. An example here is the sodium glucose transporter, or SGLT. Peripheral proteins, on the other hand, these are the receptors, the enzymes, the G protein, and the identity proteins. They do not take the entire thickness. They are either on the outside or the inside. The purpose of G protein is to connect the receptor on the outside to the enzyme on the inside. 60% of your body is made of water. 2/3 of that water is in the intracellular space, 1/3 is in the extracellular. Of that extracellular, 1/4 is in the plasma, 3/4 is in the interstitial space. Fick principle, amount equals volume times concentration, and therefore volume equals amount over concentration. No kidding. And this is how we can verify and measure each one of these compartments in your body. We give you an indicator and then we measure the compartments. How do you measure my total body water? By injecting you with heavy water and then measuring its volume. How about the ECF? By injecting you with inulin, because inulin will remain in the ECF. It will not enter the ICF because inulin is foreign to your cells. Your cells have no idea how to deal with inulin. How to measure the plasma? Give something such as Evans blue dye. It will stay in the plasma. It will not dare to reach the interstitial space. And then you know the total body water, ECF, and plasma. Then you use your brain to calculate what's the ICF is the total body water minus the ECF. What's the ISF is the ECF minus the plasma. Last thing is the Dannon equilibrium. This is before equilibrium, this is at equilibrium. At equilibrium, proteins are acting in their own self-interest. They will rig the system, making ions more abundant here. 3 plus 12 is 15. Then here, 6 plus 6 is 12. The net result is water flowing from the non-protein compartment into the protein compartment. This will preserve your blood volume inside your artery, which is known as effective arterial blood volume, to perfuse your heart, your brain, your gut, your kidney, etc. The results of Dannon equilibrium are 3 mathematical equalities and 3 inequalities. By the way, water flows from the cell into your blood vessel by 2 mechanisms. Oncotic pressure is the main one. Dannon equilibrium is the second one. Thank you for watching. Please subscribe, hit the bell, and click on the join button. You can support me here or here. Go to my website to get my cardiac pharmacology course and my electrolytes course. Share this video. I love you guys. Until next time, be safe, stay happy, and study hard. This is Medicosis Perfectionalis, where medicine makes perfect sense.